It's my pleasure. Today, it's my pleasure to introduce today's speaker, Dr. Yashi Ho, who is an associate professor of microbial pathogenesis and medicine at the Yale University School of Medicine. And Dr. Ho's research program focuses on understanding HIV-1 persistence and HIV-induced immune dysfunction using single genome and single cell approaches on clinical samples. And without further ado, please join me in welcoming Dr. Ho to share her insights on single-cell epigenetic, transcriptional, and protein profiling of latent and active HIV reservoir.
Thank you, Julie, and thank you so much for having me here. It's really my great pleasure to share some of our work. I'm very excited to show you a few stories that we have done in the past couple of years. So this is our lab. Our goal is to try to understand how does HIV do to the host cells when HIV infects a cell. Of note, the host cell transcription and cell fate decisions are tightly regulated at several different levels, not only at the RNA level in terms of transcription, but also at higher levels from chromatin interactions organizations to tightly control these gene expression. So when HIV integrates into the host genome, as we know, will that change how the host control its cellular program? And can HIV alter these cellular programs, change the cell fate, and promote its own persistence? So the goal of our lab is try to understand the impact of HIV insertional mutagenesis at different levels. So we'll start from how HIV affects these host gene control at the chromatin level. This is pioneered by my previous student, Jack Laura, and resulting in this genome research paper that he is a co-correspondent on, and then we can take a look at more details over there. So basically, host chromatin, as we know, are very, very long. They don't exist as one linear chromatin. So basically, these chromatins are arranged at the 3D chromatin structures using different things such as all these like interacting domains. So basically, just think about this. If I had a whole lot of long hair and how the host determine how to compartmentalize certain genes that you're using versus genes that you're not using, you can simply use a rubber band to tie them up. So those hairs are clustering together are arranging these things called topology associated domains. And these rubber bands that the host genome uses to group things together, these are things called the cohesing binding on the CTCF binding site. Then looking closely at these gene regulation level, you will see that here is the transcription unit, and then the transcription unit has promoters to dictate the active form versus silent form of these gene expression, but this whole transcription unit can recruit other like transcription factors through these key enhancer looping to promote these gene expression. So what happens when HIV inserts into the host genome? So the first step we look at is the openness of the chromatin, which means that if you have a tightly packed chromatin, the gene will be silenced. But if you have an open chromatin, the genes will be expressed. And this can be assessed by this method called ATAC-seq, which look at the openness of the local chromatin. Using these cell line models, having HIV integrated, we found that when HIV integrates into the host chromosome, it actually increases the openness, increasing the accessibility of the host genome. Then when we activate HIV at the HIV promoter, this accessibility increases. And when we suppress HIV promoter, then this accessibility decreases, suggesting that HIV integration, the promoter activity really dictates the host accessibility at the integration site. Then HIV jumps into integrating to the human genome. And what does it do to the gene? It's not just about open closeness of this region, because think about this. This is a promoter that brings in transcription factors. That makes it open. That sort of makes sense. But what Jack did using HIV-4C, showing that HIV can actually interact with the distal chromatin up to 500 kb, way beyond the 10 kb little HIV genome. It means that it can also actually induce the looping of other distal regions in cis to HIV integration site. And how does that help? Basically, as we see that there are these cohesion like rubber bands binding to CTCF sites to regulate host gene expression, that you can see these expressions by comparing the host chromatin interactions in the infected cells versus uninfected cells. If you see these very complicated plots, if you look somewhat symmetric, it means that they're the same. But if you look like they're not symmetric, it means that there's some different interactions. So basically what Jack found that HIV integration does not change host chromatin interactions globally. However, when Jack used enhancer loop capture, which means that if you only look at those active regions of the chromatin, that these are the places that these transcription is active as marked by H3K27 acetylation, what he found is that HIV integration into the host genome actually increases the host chromatin looping that enhances local cancer and promoter interactions. So in summary, HIV integration into the host genome is not just an addition of this 10 kb in the genome, but also increases the host chromatin accessibility, increases interactions up to 500 kb to promote HIV promoter activity. Then next, we want to understand how does HIV integration affects gene transcription and protein expression. This was spun by a talented postdoc, Timmy, who went on for another career track. So basically, back then, when we sorted these HIV-infected cells out from a participant, this is participant number 154 in this number 21 cell that we sorted out having HIV in there, he found something interesting. He found this interesting, this RNA that half of it is from HIV, the other half is the host. So why is that? Supposedly HIV splices into HIV and host RNA splices into host, but he found this chimeric RNA that is interesting, reading from HIV genome to major splice site, but splices into a host gene. Then what he found is that in this host gene called MFAT-C3, that if you don't have HIV integrated in this gene, you'll see these peaks called exons, basically just typically how genes are being transcribed. However, when in this specific cell having HIV integrated into this gene called MFAT-C3, downstream of HIV integration site, this host gene expression is increased, but upstream is totally gone. So this means that these are two promoters. HIV is a promoter, and then the host has its own promoters, like two water faucets competing with each other, who gains the control of the gene expression. In this cancer-related gene, when HIV integrates in here, now it's showing that it's now HIV that wins. It dominates over the host promoter. So now this cancer gene is under control by HIV activity. Using cell line models that we did CRISPR activation inhibition study as a confirmation, we found that normally, HIV should splice into HIV genome and host RNA should splice into the host. However, when HIV integrates into the host genome, HIV will drive aberrant host gene expression. So when HIV is integrated into cancer gene, this will change the cancer gene expression because now the cancer gene expression is under the control of HIV, no longer the host. We demonstrated that using CRISPR activation of HIV, which has higher level of host gene expression, you inhibit HIV expression by CRISPR-I, then you inhibit this host gene expression. And this can also be silenced by HIV silencing agents such as JAK inhibitor, filgotinib, and spironolactone, which has been repeatedly shown to have this effect suppressing HIV expression. By suppressing HIV expression, these drugs can also suppress HIV-induced cancer gene expression. This is important because in the past, we never thought that HIV is an oncogenic virus. It is not. Unlike other oncogenic retroviruses, such as Root sarcoma viruses, HIV is not known to cause cancer. It's other viruses that causes cancer, such as HPV, but not HIV. However, 2021, this is Steve Hughes and John Mellors. They took cutaneous lymphoma from people having HIV, asking the question, in a long-term infection, can HIV drive cancer? Supposedly not. But when they got 5 different cancer tissues from 3 different donors, they found HIV integration into this cancer-related gene STAT3, driving high levels of HIV-driven STAT3 expression. Again, this is through aberrant HIV-to-host transcription. And not only at the RNA level, they show that these cancers have active forms of STAT3 as a phosphorylated one that likely driven by HIV. So after 2021, we need to think about this change concept that HIV integration into cancer gene may cause cancer in some rare cases, and therefore suppressing HIV expression, which is in line with some of the studies done here at UCSF, may be important for long-term care. The third question we want to ask is, do host factors silence HIV expression? Supposedly, we learned this because we know from other ritual elements, such as line 1. These are ritual elements that co-evolved with humans for 40 million years. The human system had developed methods to suppress these jumping genes because line around the genome so that it doesn't continue to change a human genome. So this Pauliner and others figured out this method called Host complex, basically it's a restriction factor. If there's a line 1 ritual element, Host complex will bind and silence these ritual elements so that they won't be expressed. However, sometimes when there are these line elements that still have leaky RNA transcription, others over here found that there are other mechanisms that the host gene can't see these aberrant line transcription and try to silence them as a rescue, as a backup. These are interesting proteins that we don't really know much about, but then these are RNA processing proteins such as nuclear exosome targeting complex or poly-A tail exosome targeting complex. So basically, we as humans have developed 2 ways to silence these ritual elements at the epigenetic level or at the transcription level. Then we want to understand whether HIV can be sensed by the host this way. So therefore, we did a CRISPR screen. And this CRISPR screen uses our existing cell lines as we used in the previous paper, that 4 different cell lines each have a HIV integrated into different site, but no site that we can try to understand what the host factors do to HIV. For example, we hypothesize that there are HIV silencing factors that are binding to HIV LTR or somehow suppressing HIV transcription. If these factors are knocked out, then HIV expression will increase. So therefore, if we do a CRISPR inhibition screen to knock down these factors, and if we see increasing HIV expression, then we suppose that these are factors that are silencing HIV. So with that, and through these 4 cell lines to create biological replicates and using these methods such as MAGIC or LIGER, we found a list of genes that can silence HIV. Then when my student Savannah presented this to me, I was like, these are just the alphabet soup list of genes, and what do they mean and how do we learn? The answer is, if you plot out all these genes in RNA biology, we found something interesting. So first, we found these family of genes called SAFB. Basically, there are 3 SAFB proteins in the SAFB family called SAFB1, SAFB2, and SAFB-like protein. We got 3 out of 3 in this CRISPR screen. And this is an interesting protein because it has a DNA binding domain, RNA binding domain, and a protein binding domain, providing it a perfect way to either epigenetically or transcriptionally interact with exogenous DNA RNA or as a scaffold by protein interactions. Surprisingly, back in 2018, someone did a study trying to identify line 1 suppressive element. They found these Host complexes, but they also found SAFB. How does SAFB do to HIV is still under our investigation. And second, we found these genes that seem to have unknown names and funny names. We don't know what they are, but turn out these are the exact genes that controls line 1 element inferior leaky expression from the line. So these are genes that are supposed to control leaky line expression, and we found that these are also genes that may potentially silence HIV. So these are very interesting to us. And finally, we want to try to understand how HIV-infected cells survive, persist, and proliferate. Basically, from chromatin interactions, transcriptional interactions, we want to see how these differences that HIV causes changes cell fate decisions and therefore HIV persistence, which is relevant to our pursuit in how to find a cure and developing HIV cure strategies. So why can't antiretroviral therapy cure HIV infection? Many people, even scientists, still ask me, people just take ART and viral load is perfectly suppressed. And why are you studying HIV cure? This is perfectly suppressed. The answer is antiretroviral therapy is indeed very effective. It does stop new rounds of infection. However, ART does not kill infected cells. As early as 3 days after infection, there are lots of millions of HIV-infected cells that already establish HIV reservoir in the body. That does ART kill HIV-infected cells like penicillin would have killed bacteria? The answer is no. See, ART is not cytotoxic. You can have a whole tube of ART and add that to your cell culture. Cells are perfectly fine. ART is not like penicillin killing bacteria. ART is not cytotoxic, and therefore ART does not kill HIV-infected cells. And second thing people ask is, oh, there are people who are taking antiretroviral therapy, but they have low-level viremia, for example. They can have these, viral blips being detected, but you said ART is suppressive. So why is it? And I would like to ask you to read, for example, Jonathan Lee's recent paper on profiling low-level viremia, paper recently in Nature Med. Basically, they're just these people taking ART perfectly, every day with no drug resistance, but still they can still detect plasma viral load. And why is that? This is because ART does not suppress HIV promoter. Antiretroviral therapy inhibits viral enzyme activities, but they're not CRISPR. They do not suppress HIV promoter activity. And HIV-infected cells are still expressing a lot of HIV RNA and proteins. Basically, ART can suppress these protective infections. Some cell will return to a latent state, and this is a time that we don't detect any plasma viremia. However, stochastic antigen stimulation still happens every day. For example, these cells can be specific for SARS, can be specific for HIV, CMV, rotavirus, anything. So during the stochastic antigen stimulation, HIV promoter will be activated, inducing RNA transcription, protein expression, and thus inducing chronic immune activation. This is pretty much like how I need to weed these like dandelions in our backyard. Basically, you can use a lawnmower to clean them, but the roots are still there, just like these integrated HIV proviruses. And even under ART, they can still be expressed in creating chronic immune activation. And what is even worse, these HIV-infected cells not only stay there over time, they proliferate from one into many, even under ART. As we know, ART does not stop proliferation, does not stop HIV promoter. And not only these cells persist, but they also proliferate, creating an increasing barrier to HIV cure. Not only that, if we want to understand these HIV-infected cells, imagine what we do with cancer. For example, if you want to study cancer, you want to understand these cancer cells in every aspect, for example, in their mutations, their drug resistance, are there markers that we can study or target cancer cells. But HIV-infected cells are extremely heterogeneous. They can be having different integration sites, including about 90% in introns, about 10% heterochromatin. They can be intact or defective, especially only 1% of the provirus that we sequence are intact, but 99% are defective in all different ways, such as packaging signal deletion, large internal deletions, and hypermutations. And the HIV RNA expression is also heterogeneous because of stochastic expressions such as tag-mediated stochasticity and many other reasons. So HIV-infected cells are just so heterogeneous and hard to understand. So this is our last current view about the top questions that we would like to pursue. That what prevents us from developing an effective HIV cure strategy? Supposedly, HIV integrates into the human genome in CD4 T cells, and when they are latent, the immune system cannot see them. So one of the current strategies that field is using is called the shock and kill strategy, basically using latent reversing agents to induce HIV expression. So then these cells can either die of viral cytopathic effects or be killed by the immune system. But no, sometimes these cells just don't die of viral cytopathic effects. Why? And the second question is that sometimes these cells are just resisting CD8 T cell killing. Why don't they die of these immune clearance? And third, these cells not only persist, but also proliferate into many. What drives their proliferation and how do we stop them? And fourth, these cells are so heterogeneous. Imagine when we're studying breast cancer, colon cancer, if we know a different mutation, we want to target them with a different immunotherapy. If we have not understood fully about the heterogeneity of HIV-infected cells, how do we effectively target them? And this is why our lab in our field has this 1 in a million problem that only 1 in a million CD4 T cells contain these HIV RNA positive cells. This is so rare to find. It's like needle in a needle stack, because not only is like needle in the haystack, but there's no marker for HIV infected cells, that we cannot distinguish them from uninfected cells. Unlike breast cancer, you can take them and sequence them. We just draw blood. We don't know which cell is positive HIV or not. So this is like a needle in a needle stack. And third, these HIV CD4 T cells are actually very heterogeneous. Can be Th1, Th2, Th17, central memory, effector memory, activated, exhausted. So for us to understand HIV infected cells in the middle of these heterogeneous CD4 T cells, it's very difficult. Just like how to find this rare, high resolution understanding of black hole in the middle of the galaxy. And that's why we and others, including Nadia, are trying to use single-cell multiomic approaches to profile the heterogeneous CD4 T cells and find that rare HIV-infected cells. Because if we simply just draw the blood and try to find markers and correlations, we're looking at 99.9% of the uninfected cells, and that does not tell us what's really happening in HIV-infected cells. Therefore, we love to pursue single-cell multi-omic methods, and I hope that by the end of the talk today, I can convince you that we're using this method, not just because it sounds fancy, or everyone is doing it, or it looks beautiful, although I don't know what that means. I found signatures, I found new cell types, but having no meaning. I hope I convince you that it is because the problem that we're solving is it's a heterogeneity and rarity of these HIV-infected cells. And I hope that what we found can be mechanistically validated to create biological or therapeutical insights. And this is a different way of approaching these questions, because in the past, we typically use flow cytometry or other ways trying to target our favorite markers to identify HIV-infected cells. However, now, because of technology advancement, if we're able to look at single-cell RNA-seq, which means genome-wide profiling of all the genes up and down regulation, I hope that we can have a better understanding of these heterogeneous HIV-infected cells. Initially, about 6-7 years ago, this is how we started, that we tried to identify these HIV-infected cells for single-cell analysis, but there's no marker. So we activated these cells with PMI anamycin, so then HIV will start to express HIV RNA. Then we can design probes to bind to these HIV RNA using HIV RNA surrogate to sort out HIV RNA infected cells for single-cell analysis. Similar approaches have been done by Lee and Cloven and Michelle Nielsen-Zweig, or Dan Kaufman and Nicholas Shaman, trying to use HIV RNA or protein expression as a surrogate to find those HIV positive cells. So while this is interesting, but after activation, these cells are no longer what they were in vivo. So while we can understand certain signatures, this does not represent what's really happening in people having HIV. The past year has been a very exciting year, that there are different groups trying to understand HIV-infected cells without in vitro activation. This is Ian Clark and Eli Boris using PCR-activated sorting to sort HIV-positive cells and then sequence them. And basically, this is an elegant method. However, the PCR-activated sorting of HIV DNA-positive cells destroy the RNA, so they can only look at HIV transcriptome about 100 cells together, not at a single cell level. So this does not resolve the heterogeneity. Matthias Lichtenfeld used FEP-seq, which is the Mission Bio-based system, to do targeted amplification of HIV proviral genome, so they can look at the intactness of the HIV proviral genome and with paired protein expression. However, because of this targeted amplification of DNA also destroyed RNA, this method also is not able to capture RNA-seq at the genome-wide scale. Then Vincent Wu and Michael Betts used ASAP-seq, which is using transposase, using ATAC-seq, to see if transposase happen to bind to HIV DNA, then they can identify HIV DNA positive cells with paired protein expression profile. However, this method at that time still could not pick up RNA expression. So we're showing that there are two methods, Excite-seq and Dogma-seq, and my student Jack and postdoc Yulong used trying to identify HIV positive cells with their DNA, RNA, and protein profiles. We collaborated with Dr. Ann Dewar at Fred Hutch, who runs a clinical trial of the Sabes and Merlin cohort. Basically, this is a village of people at risk of HIV infection. They do prospective testing of HIV tests. So basically, these are people who are at risk. So they got tested for HIV positivity once a month. So when these people turn out to be positive during viremia, they collected blood samples and started antiretroviral therapy. After 1 full year of antiretroviral therapy with documented undetectable viral load, they collected another blood sample so that we can compare the HIV reservoir dynamics from the same people during viremia and after viral suppression. So with that, here is our student, Jack, who very excitedly read a bioarchive paper and just trying to say, we can try to do this. This was the paper describing ESIC when it was in bioarchive, later published in Nature Method, developed by Peter Smippert. Basically, this is an RNA-seq-based method, and RNA-seq is actually easy. So you just need to couple 3 things together: the cell, the reagents, and a barcode into one same bubble. So in this bubble, for example, this is what 10x commercialized can have these 3 things coupled together at an efficiency of 90%. And this is why this platform is so far dominating the field, because the earlier versions, such as InDrop and DropSeq, are only 5% incorporation capacity. So with that, you can capture the cellular transcriptome and define which cells they are, such as Th1, Th2, Th17. The elegant part of this method, just like CytSeq, is that they can have an antibody that is tagged with a DNA tag. So instead of PE50 that you would typically use for your fax antibody, so this antibody is now tagged with a DNA tag. So now after sequencing, we can see what are the proteins that are bound to the cell surface, for example, CD45RA and CCR7. These are markers that are, for example, CD45RA are splice variants that cannot be distinguished by transcriptome. Then we can faithfully determine their surface protein and their immune phenotypes, such as memory, central versus effector memory. We map the RNA transcriptome to HIV RNA, so we can see which cells are RNA HIV positive. Then we can also map to T cell receptor sequence, which means that there are these T cell receptors that dictate the antigen specificity of each cell. So when we see different CD4 T cells that have the same T cell receptor sequence, as you see in these dark colors, it means that these are cells from one same mother cell that proliferated into many, because T cell receptors are unique for any single CD4. So when you see different T cell receptors, different cells having the same T cell receptor, it means that it has undergone clonal expansion in vivo. So these are the cells that are clonally expanded, proliferated most in vivo. Then because we have the transcriptome data on these clonally expanded cells, then we can try, these are cytotoxic CD4 T cells that are the most clonally expanded cells in vivo. Next, Jack compared what are the markers for HIV RNA positive cells versus negative cells. Then just doing these volcano plot, which means that these HIV positive cells have increased expression of these genes at very good significance. He found that these are genes and these are cells having activation markers, such as HGDR, which is known to the field, such as what Sarah Palmer previously characterized. HIV positive cells may express exhaustion markers, such as PD-1, CTLA4, LAG3, and TIGIT, which is also known in the field, as published by Latent Shaman. But what Jack found was that HIV positive cells actually upregulate perforin, coenzyme K, CCL5, coenzyme A. These are like cytotoxic T cell response genes. And this was not known before. This is exciting to us. Wow, now is it HIV enriching cytotoxic CD4 T cells? That is interesting. We next want to understand the T cell clonal expansion dynamics in these HIV people living with HIV. This is because we wonder whether the CD4 T cell expansion dynamics is dictating the HIV infected cells within these CD4 T cells. So basically, each CD4 T cell has a unique T cell receptor sequence. And the job is that when antigen presenting cells present an antigen, such as SARS or HIV, to these CD4 T cells, these CD4 T cells will be activated and proliferate. HIV loves to infect these activated CD4 T cells because the recruitment factor such as MHC1 is now removed. And now HIV is integrated into the host genome. Just of note, this T cell clone is not fully infected with HIV. It can be just one or part of all of cells infected with, which not 100% of these are HIV infected. When the antigen stimulation continues or recurs, these CD4 T cells corresponding to this antigen will proliferate, same as these HIV-infected cells that are within the same the T cell clone. So we want to try to understand this is called HIV-positive T cell clone, although only a proportion of the clone is HIV positive, but we hypothesize that the same antigen stimulation will drive this T cell proliferation that also drives these big HIV positive T cell clones. Then with that, we can look at three different things: clone size, clonal expansion dynamics, and clone phenotype. So first, we found that in HIV positive T cell clones, compared with HIV negative T cell clones, HIV positive T cell clones are always bigger in clone size, which means that there are many cells that are proliferated already much more in vivo than uninfected T cell clones. And second, we found that these HIV positive T cell clones persist over time. What does that mean? It means that during antigen stimulation, the T cell clone will proliferate, but when the antigen is gone, such as SARS is recovered, then the T cell clone will disappear. But HIV positive T cell clone are peaking here, which means that they persist over time. If you found 1,000 cells during viremia, you still found that 1,000 cells during viral suppression. And you can track this because they have the same TCR sequence. And these TCR clones having the same TCR sequence actually persist with the same phenotype. They are cytotoxic effector memory cells during viremia and after viral suppression. They're already polarized, and that did not change over time. By looking at the T cell clonal type, we found that these HIV positive T cell clones, that 75% of them are in the same cell subset called cytotoxic Th1. So we found that it's the cytotoxic Th1 T cells that are enriched with these proliferating T cell clones. Then we found that these T cell clones do upregulate granzyme B, granzyme H, granzyme A, and perforin, and we used flow cytometry to validate this result using the SABIS cohort where we collected our samples in a separate cohort in Philadelphia, having a different ethnicity, different age, and different ART regimen. Then my friend Brad Jones told me, hey, Yachi, did you look at SERPINB9? I was like, what's this? Basically, SERPINB9 is a granzyme B inhibitor. Basically, in all cytotoxic immune cells, such as cytotoxic CD4s, NK cells, and cytotoxic CD8 T cells, they are all making lytic granules, such as granzyme B, to kill target cells, such as cancer. However, to prevent cell-inflicted injury of these lytic granules to hurt the producer cells themselves, these cells express SERPINB9 as a granzyme B inhibitor to prevent cell-inflicted injury. Then we found that indeed, HIV positive cells have higher level of SERPINB9 expression, and that's maybe why HIV-infected cells can persist, because HIV infecting different CD4 T cells may be random. But if you happen to jump into cytotoxic CD4 T cells, these are cells who love to proliferate. And if HIV infects these cytotoxic CD4 T cells, and when cytotoxic CD8 T cells want to kill them, you have the weapon, I have the weapon. You have the shield, I have the same shield. So we propose that these HIV-infected cells, when they infect cytotoxic CD4 T cells, these cells may resist cytotoxic CD8 T cell clearing. And when these antigen stimulation continues, HIV-infected cells continue to proliferate because these are the cells that proliferates most in vivo. The study have been repeated in different methods, in different cohorts, in different groups. This is Michelle Newson-Spike, this is Rosmi Thomas. They both found that HIV-positive T cell clones or HIV-positive cells both upregulate granzyme A, granzyme K, and perforin, suggesting that while you feel like these single-cell transcriptomics can be just random signatures, we do find that different groups of people identify similar results, cross-validating each other. Then people asked us, hey, Yachi, you found these transfusion active HIV-infected cells. How about those latently infected cells? What do they do? I say, no, I don't know, because Jack used HIV RNA as a surrogate to identify HIV-positive cells. If they're DNA positive, but RNA negative, that they're latent, I can't capture them using this exact-seq method. So we use a different way, but we use the same cohort and recruited in the Sabius and Merlin cohort led by Dr. Ann Dewar, but using a different method this time called dogma-seq. uses a method that was described in Nature Biotech by Aviv Regev and Peter Smigberg, but then this is adapting it to HIV platforms. So basically, dogmacy is called dogmacy because it helps us to understand the cellular state at DNA, RNA, and protein level through the central dogma of molecular biology. Basically, using ATAC-seq, we can look at the epigenetic regulators of these cells. Using RNA-seq, we can look at the cellular phenotypes. Using CytSeq, now expanded to 156 surface protein markers, we can understand the cellular markers or their surface protein expression profiles. Now, we can use transposase to detect HIV DNA. We also can use our transcriptional mapping to identify HIV RNA, and therefore we can identify HIV DNA positive cells and HIV RNA positive cells separately with their combined epigenetic, transcriptional, and protein expression profiles in the same single cells. These are typical plot that you will see, very beautiful, having clusters here and there and here and there, and you wonder, how do I find these findings of Th1s here and here and proliferation there and there? Fortunately, Rahul Satija published Weighted Nearest Neighbor. This is in Sirah 4. Basically, this method integrates all different modalities together using ATAC-seq, RNA-seq, and surface protein expression. Now, this plot shows all different modalities in the same single cell. Basically, all the cells that you see on this plot has all three modalities in each single cell together, telling us how these cells are differentiating and their different phenotypes. This is a very difficult method because if you think about how effective can we detect HIV in this system, the answer is it is very hard because HIV DNA is just 10 kb, but human genome is 6 times 10 to the 9th, like a 6 billion if you think about it as diploid. So it's just about 0.001% of the human genome having HIV. So for the transposase in ATAC to capture HIV DNA is very, very sparse. We try to sequence double the depth as Chanik suggested, but about roughly 28% is the highest capture rate that we can get. For RNA-seq to capture HIV RNA is actually easier. Well, not easier. It's because HIV RNA can be expressed at multiple copies per cell, but HIV DNA is likely just 1 copy per cell, and therefore RNA-seq can capture HIV RNA differently. So again, we sequence deeper than what is being suggested, and then if we find like no more than 2 reads per cell, which is our detection limit. So if we call it HIV RNA negative cell, we call it like 2 out of the 50 reads that we sequence around 0.04% of the cellular RNA. If it's below this level, we call it HIV RNA negative, although it can still be lowly expressed, not captured, but this is the threshold that we use. And we define the sensitivity and specificity by looking at these mapping on infected cells, and we capture 0 to confirm that this is a specific method without artifacts generated by deep sequencing. So with that, we're able to capture HIV DNA and HIV RNA. You can see their landscapes that HIV DNA is more captured at the promoter region, consistent with the mammalian cell genomics, that promoter regions are more accessible and open. So with that, this is the first time that we can identify HIV latently infected cells versus transcriptionally active infected cells in the in vivo state without ex vivo stimulation. And this is the first time that we can identify single cell transcriptome of latently infected cells. This is during viremia and after viral suppression. So we identify a total about of 500 cells together. So we first look at their ATAC-seq profile, which means that what are the transcription factors that are dictating the cell fate of these cells, such as latently infected cells versus transcription active HIV-infected cells? Are they different? In the past, we thought that latently infected cells should look just like uninfected cells because they don't express anything. But we found that actually latently infected cells are having increased transcription factor activities, RORB and RORC. These are Th17 directing transcription factors. In contrast, the transcription active HIV RNA positive cells have other transcription factors dominating their transcription activity, such as JUN-FOS, BAF, and BOC2 and IRF. Next, we look at RNA-seq. Basically, what are these transcription profiles of latently infected cells? Again, in the past, we thought that latent infected cells should look just like uninfected cells, but we found that yes, there are still differences that we can capture. For example, we see granzyme A that we found in our previous study. Again, granzyme A and granzyme K reproducing what we found, that these are indeed markers that can enrich for HIV-infected cells. But transcription latent and active HIV cells seem to have somewhat different markers. Then we ask, can we find markers that are seen both in the latent infected cells and active infected cells, so that we can try to validate them and use them as markers for HIV, like infected cells? We found these A genes. Some are our old friends, such as type 1 interferon responses during viral infection makes sense, and cytotoxic T cell response genes make sense. This is what we saw last time. These cells may have increased IL-2 production and increased integrin alpha-4 expression. This is previously described by Nicholas Shaman. These all make sense. But we found this interesting gene called IKZF3, and the protein name is called ILOS. This is the Ikaros link finger transcription factor that regulates lymphocyte differentiation. And what is it? We just found it. After some reading, we found that IKZF3 is helping Th17 and TFH differentiation. In some studies, if you have a dominant positive mutation, then you can increase BCL2 expression, increase cell survival in the B-cell lymphoma model in mouse. Then HIV integration into IKZF3 has been reported in different studies. So we found this interesting. But what does IKZF3 do in HIV-infected cells? We'll try to do a WGCNA-based module analysis, basically trying to find, is there a way to distinguish HIV RNA positive cells versus negative cells? And this is important because he wants to find out what makes these RNA positive cells survive. Given that they're actively transcripting HIV RNA, making proteins, they should die of viral cytopathic effects, but they don't. But instead, these cells may still express Ki67, the proliferation gene. What are the genes that are co-expressed with this proliferation module? He found BRC5, which is surviving. Then Matthias Lichtenfeld had a paper on IL-21 and IKZF3. So this is interesting. So we did an in vitro validation that we took activated CD4 T cells, infect them with NL43 or other clinical isolate just to gate out HIV positive cells versus HIV negative cells. This is positive, this is negative, and try to see if their IKZF3 expression level is different. The answer is yes. HIV positive cells have higher level of IKZF3 expression, and these cells having IKZF3 also have higher level of Ki67 expression, suggesting their increased proliferation potential. And finally, when we use the seq to look at different surface protein expressions, we found that these HIV positive cells are polarized in Th1 or Th1 star, which means Th1/Th17, such as CCR6 markers, as well as other homing markers and activation markers. Then by the end, with all these fancy, beautiful multiomics, what do we learn? We did learn that HIV-infected cells are very heterogeneous. Yes, but how does it help us to understand how HIV persists? So Yulong pulled all these 533 HIV-positive cells together and tried to use ATAC-C to find their cell differentiation trajectory. Given their heterogeneity, are there shared pathways these cells are differentiating into? He found that during acute infection, there are these IRF interferon response genes, like STAT1, STAT2, that leads to activation markers and survival programs. Some cell will die of viral cytopathic effect. Some cells will differentiate into cytotoxic CD4 T cells, as we show through EOMs with these cytotoxic T cell genes. And some cells, because of TNF and June-Phos transcription factor activity, will differentiate into either T17 or certain homing genes. So we're still trying to understand how does AP1 and TNF shapes the differentiation of these HIV-infected cells, but there is this trajectory dictating the cell fate of HIV-infected cells. And finally, we've been looking at HIV-infected cells in the blood, but what about the tissue? In the past, we did not study tissue because these samples are so hard to get. Now that when we have access to tissues, what do we do? Indeed, we can just grind up the tissues and try to understand the single-cell interactions, as we did, which we're actually doing, and this is great. However, the caveat for this is that this lacks the interaction part between HIV-infected cells and other immune cells. The typical commercial platform, you will have Visium, for example, trying to understand spatial transcriptomics at 55 micrometer resolution, but that's a mixture of cells, which does not recapitulate what happened at the single cell level, because a single cell is about 10 micron for a lymphocyte. Therefore, we use STPseq developed by Rongfan's group, and here at Yale, trying to understand these tissue and spatial transcriptomics at a closer to single cell resolution. And our postdoc Amare is further using SeqScope, another platform, to look at these HIV positive cells further down at subcellular resolution. As an example, this is Kristen, who spent her COVID time in Rongfan's lab. Basically, Tom Hope sent us HIV positive tissue samples and in frozen sections, and Kristen will cut them for histology, spatial ATAC, and spatial RNA-seq. So using these microfluidics that she made in-house in her hand, she can detect SIV DNA using spatial ATAC and SIV RNA using spatial RNA-seq. And you can see that what she captured represents the different splice forms of SIV RNA, and she can identify authentic SIV DNA at the spatial context. She's still analyzing. So finally, in summary, how do we understand HIV reservoir better and faster? Back in 1984, when HIV was discovered, we're trying to understand what are HIV virions, and we try to understand their viral landscape using RNA sequencing of the plasma or the virus. 1997, Bob Slicano, Tony Fauci, and Doug Richman discovered the HIV reservoir. Now, there is a wave of about 20 years. People are trying to understand the reservoir, trying to measure the reservoir to guide clinical trials. Then people try to understand how many. And myself included, we're trying to understand the HIV DNA proviral landscape. Now, because of the advancement in single-cell technologies, we can now look at why and how HIV persists by looking at their cellular landscape. And therefore, we've been moving away from viral RNA, viral DNA, proviral DNA, now over to the cellular landscape to resolve the heterogeneity of HIV-infected cells. What we've learned in the past years that is different from what we learned 10 years ago is that in the past, we study latently infected cells because transcriptionally active HIV-infected cells would just die of viral cytopathic effects. So don't study them because they will die tomorrow. However, by papers from ourselves and Matthias Litterfeld, we found that these transcriptionally active HIV-infected cells, they actually survive, persist, and proliferate. So for us to eradicate HIV, we should consider both of them, both latently and active HIV-infected cells, that we should extend our scope beyond the latently infected cells. The second thing we learned is that in the past, we dichotomously divide C4 T cells as resting versus activated, because we assume that resting cells has latent HIV, activated CD4 T cells have HIV that will just die soon. But now that we understand transcriptionally active HIV-infected cells don't necessarily die, and we realize that dichotomous dividing CD4 T cell into resting and activated no longer captures the heterogeneity of cytotoxic T cells, Th1s, Th17s, and TFH. We hope that we can now use these different tools to understand HIV reservoir better. Finally, I hope I convince you that I'm not just showing you some beautiful, colorful plots. We identify IKZF3 that can help these HIV-infected cells to proliferate, and there are drugs such as lenalidomide that can inhibit IKZF3. We're actively testing this. We also identify SERPINB9, which is the granzyme B inhibitor that may help HIV to resist immune clearance, and we're also testing SERPINB9 inhibitors. And by the end, we have not cured any person in my hand. Why do we study these things that seemingly fancy, but there's no cure strategies involved yet? This is because we believe that we need to know better about what we're fighting against. We are building a map. The better the tools we have, the better resolution of our understanding of something unknown, such as the big wide universe, or such as the heterogeneity of HIV-infected cells. We are not yet satisfied to find one marker as the magic bullet. I think we're not there yet. We are still exploring more to know what we can use to understand HIV. And these are methods that can help us to understand HIV persistence over time and space, and using anyone's favorite interventions, and hope that these are methods that can be applied to all different strategies, not necessarily just our group. So with that, I would like to thank our study participants, funding sources, and our collaborators in our lab. And the final project was mainly done by Yulong here, our postdoc, and we have other studies going on. Thank you.
Well, thank you so much for the great talk. We have time for questions. So if people want to unmute themselves and ask their questions, it's the time. So go ahead, Nadia.
Hey, Yachi, nice seeing you.
Hey.
Talk as
Thank
usual.
you. Yeah,
I have a couple of questions. So the first is when you were presenting your Serpent B9 conclusions, the way you had presented it was in the context of HIV preferentially infects the cells that are granzyme B positive, but at the same time have Serpent B9, therefore they're protected. But could it also be the case that HIV infects a cell that doesn't have that signature, but then kind of induces that kind of state to protect itself? And have you kind of looked into how you might test or distinguish the two possibilities? That's my first question.
Can I answer this first? So this is a very good question because there are three possibilities we're thinking about. One is preferential infection. Mm-hmm. Second is infection is the same across, but preferential persistence. And third is that you infect the thing, but you shape it. For example, if I infect the TCM or naive cell, but then I shape it into a cytotoxic CD4, for example, there are these three hypotheses that we're thinking of. We don't know about preferential infection yet. I am guessing preferential persistence, that infection can, when we look at infected cells, any cells can be infected. Naive cells can be infected. Th17, Th2 can be infected. But I would say that I'm guessing because we find they provide preferential persistence because they proliferate better and because they may somewhat resist immune clearance. Whether that's HIV shaping it into a different cell fate, we actually don't know yet. That will be fascinating to test, but we don't have evidence yet to say so. But we do have a PhD student trying to figure this out. She just presented last Friday, but we feel like we don't have enough premise to call it. If we are able to prove it, that would be interesting, but I don't have data.
Cool, thanks. And then my second question is, have you compared the infected, so you have your cohort analyses, you have the before ART and then after ART. Have you compared the infected cells, the HIV RNA positive cells or DNA positive cells, however you want to define, have you compared it between in the context of viremia versus suppression to try to see whether there are unique signatures in the actual reservoir cells as opposed to the ones existing during viremia?
So so actually the majority of the data we have HIV RNA positive cells, the DNA positive cells, mostly from viremia, as you can tell that the cell numbers are bigger. So when we wrote the paper, most of the data was driven by the viremic tempo. Then you're like, this is not reservoir. Yes, I agree. So what did you do when you look at the reservoir? There's so few cells that reviewers really hated us to make any conclusions. So we made some iffy potential, yada, yada, but we didn't addressed it fully. We do separate them for analysis, but we do not want to claim one factor being the dominating. I would say the cytotoxic T cell markers are repeatedly seen in both studies. June phos was previously seen both in ours and microbat studies, so those are for sure. IKZF3, I would guess, is more about a viremia marker that we see some increasing virally suppressed, but that's just to be confirmed. But in the short answer is, we will need to sequence much more virally suppressed samples to get a more confident.
And what about the cytolytic signature? Just curious, did you see it in both, or is it too few cells to look at?
We saw some, but there's still too few. So if you look at the clones, then yes, we do see them. But then reviewers hate our dogma-seq and clonal analysis, so we just removed that part. But okay. We actually saw those. Yes.
Thanks, Yachi. Great talk.
I don't know if there's any other question. I have one. Oh, Melanie, go ahead.
No, Julie, go ahead. You go first.
Okay, so I was wondering, it's about the persistence of the clonally expanded cells with HIV. And so you said that it might be because they proliferate better and they can resist to killing with Serpent-9b. And I was wondering if you know if it's if those markers have been also shown in other cells infected by other viruses that induces clonal expansion like CMV or I don't know.
And when we look at, for example, markers like granzyme B, I found a remote paper saying that if you have HCV infected cells in the liver, they may also resist killing by upregulating this. But the paper looks somewhat iffy, so we don't know. what we know is about the cancer field. There's a Cell paper about 3 years ago by someone in Boston saying that cancer cells can upregulate SERPINB9 and resist killing. So that is what the cancer people had already found. For CMV, I don't specifically see that increase. I think for CMV is what Rick Halbert, Danny Dweck found about like 20 years ago. They say that CMV specific cells, they upregulate CCL5 that make it more like just it's a ligand and it binds to CCR5, so it makes it more resistant to infection, but we still see them infected. So the short answer is we only look at HIV infected cells. We don't know about viral infected cells, but this analogy was seen in cancer.
Thank you. Melanie?
Yeah, quick question. I'm really struck by the CD4 CTLs. Can you tell us a little more about them? Are they all the same? Are there different subtypes? Are they all highly transcriptionally active? What I don't know.
Yeah.
The deal.
Let's see. So it's actually an interesting question, because when we publish a paper, there are people shouting at me saying that these CD4 T cells are helpers. Why do you say they're cytotoxic? So that I didn't invent the term. So I tried to pull out, there is an investigator named Susan Swain at UMass. She has been working on cytotoxic CD4s for a long time. She has some review articles and describing it very nicely. Then we read in the cancer field about a couple of years ago, there's a bladder cancer paper saying that cytotoxic CD4 T cells are the major effector, not CD8s, to kill these bladder cancer cells. They express granzyme B and granzyme K. It's totally MHC2 dependent. You block MHC2, the killing is gone. So I would say these are less focused, but indeed exist cell types in previous method by immunologists and by the cancer field. Are they transcription active? I would guess so, but I don't have a direct answer for you. For example, are they having higher RNA content? I'm not sure, but I would assume we capture a whole lot, and these are highly expressed genes. So we do see good signatures such as granzyme B, granzyme A signature. So I would assume that they are quite transcriptionally active. Unlike naive cells don't express much, for example.
I was muted. Sorry, that's the cells that harbor the transcriptionally active reservoir.
Yeah.
No?
Yeah.
All right. Okay.
Yes, go ahead.
Can you hear me? Good. Thank you for the talk. On the first part of the talk where you were discussing these hybrid RNAs where there's a splicing between the genes, I have two questions.
Yeah.
The first one, do you see an effect on the upstream promoter? Because it seems that the scale is different, or it's like the regular gene, the standard gene keeps expressing the same, and then you have this add-on of these new splice variants, or there's an effect?
Not the upstream. So first it's location, and next it's location and orientation dependent. Basically, splicing can only happen one way, not backwards. So then so the host promoter is upstream and the HIV promoter is here. You can only splice down. And second, unlike some other retroviruses that can read through, what we find is that HIV does not read through outside of the transcription unit. It's within the transcription unit. It doesn't add on reading into like a downstream thing, just within the transcription unit, and outside is not affected, just the exact gene where HIV is integrated.
Good. And the other question is, do you think there's anything specific about this gene that makes this happen, or it's a common feature that whenever HIV gets infected, you get these hybrid mRNAs?
We actually see these hybrid RNAs quite often. Like for example, in cell line models, it's quite easily seen because the cell line models. So we have some, and then Emory, they like Svenos group, they also have some cell lines, and then you look at, for example, JLADs, you see them. We sequence and you see them coming out. So these splicing are commonly seen. It's just that when it's in any like a housekeeping gene or like an important gene, it just happens, but it doesn't cause anything to the cell. But when it's integrated into cancer genes, such as the VAT1 we tested in our cell line models, that increases cellular proliferation.
Okay, I think there is no more questions. So, well, thank you again for the great presentation and discussion. And for some of us, we're going to meet you in like 30 minutes for the lunch with trainees. And for everybody else, thanks for being here. And thank you again.
Thank you. Thank you so much. Thank you for coming. Thank you.